is there any organism which can remove metals from such complicated e-waste, and how? So here comes the terminology called as bioleaching. So the remediation of e-waste, it can be done by bioleaching. That's what actually we have approached. This was the approach from our side. So as you know, what is bioleaching? So this is a extraction of metals from the solid matrix. Bioleaching, some of you might be knowing ki, the mining areas, the mines from where we are extracting the metals. So the metal extraction is economical if the metal is in a higher concentration. Sometimes if the metals are in lower concentrations, then the existing methods would not be appropriate, would not be good to extract the metals from economical point of view. At that time, bioleaching is used. So here in bioleaching, nothing but the organisms, microorganisms, they are being used. So these are the ore samples, pyrite, chalcopyrite, pyrrhotite. So basically removal of metals by using microorganisms. So here two mechanisms they are there, chemolithotrophic leaching and heterotrophic leaching. So let's see it one by one. In chemolithotrophes, as the name suggests, carbon source is CO2 and litho, inorganic source is an energy source. So ferrous ion here in this case is an energy source. Mechanism is Fe3 oxidizes the metals. So Fe3 ions, ferrous is converted into ferric during the growth, and ferric ions which are generated, they are working as a leachant. This is a electron transport chain of the organisms. You can see over here this ferrous, they are oxidized, they are giving electron, and then the NAD is converted into NADPH2. Okay, this is again another mechanism. I'll not go in detail of that. So here you can see that this reddish color is because of the organisms, bioleaching iron oxidizing organisms. These are some electron microscopic images for the chemolithotrophes. Chemoheterotrophs, these are using organic compounds as a source of carbon and energy. And here the acid which is produced by the organisms, this is leading to the leaching. So metals are dissolved in the acids synthesized by the organisms. So some examples of here are some chemolithotrophic examples. Here are some heterotrophic examples. As you know, as we have discussed, the mechanism in this case is different. The mechanism in this case is different. Yes, this is what we can say, dump leaching on a slope. So the in situ, the word in situ and ex situ, we have encountered in the very first slides of this presentation. So there is an in situ leaching. In situ leaching, in this technique, the ore is not moved from its geological setting, with the advantage that excavating costs can be saved. See, so as to make it economical, in situ, wherever the this thing is present, the ores they are present. So there only the pipeline have been attached in this way, so that the liquid will be spreaded throughout, and it would be finally collecting the leachate containing the metals. This is the in situ leaching, in situ bioremediation. Yeah, here the approach I want to share with you people. See, generally, whenever we are using microorganisms, so at that time, generally it is how the consortium is built up. You might be aware of the consortium built up. So what we are doing is generally we are taking separate isolate. Number 1 isolate, number 4, number 19, number 25, like that. These are the isolates which we have given good results. So what we do generally is we are taking these separate isolates, we are mixing it together, and we are calling it as a consortium. Fine. This is how we are doing it generally. So what I want to tell you, here we have used a different approach. Whatever the natural organisms are there, we have enriched that from different ore samples from the pyrite bauxite. So these organisms from the pyrite, we have enriched all organisms from the ore sample. We have enriched all organisms. Rather, we have kept the ore sample as it is in the medium, and whatever organism wants to grow, we have allowed them to grow. Same is with the pyrite, same is with the bauxite. After that, we have mixed these cultures together. Whatever the mixture we have obtained, we have mixed together. And this we have called it as a mixed bacterium consortium. Mixed bacterial consortium. And now, this mixed bacterial consortium we have used in our process. The logic behind that was whatever the organism is more compatible, so that organism only will survive. Okay, so here we have used a different approach for consortium buildup. Generally, the approach which I have previously discussed is called as bottom up approach of consortium buildup. Here, in our case, we have developed top-down approach of consortium buildup. Fine. So here in this bioremediation, we have used a top-down approach of consortium buildup, and we have allowed the organisms to interact with the waste material. Most importantly, see, all of the microbial processes, they are generally using sterile conditions. It should be sterile, the reactor should be sterile, the process should be aseptic, like that. In our case, we have throughoutly used non-sterile conditions. Throughout, we have used non-sterile conditions because, see, what's the risk in keeping the non-sterile condition? There are chances that contaminants may enter. So in our case, we were not bothered about the contaminants, because even if the contaminant, see, basically the media was too acidic, it was, the pH was around 1 to 2, and the second thing is there is no any organic material for consumption in the media. It was totally inorganic. So in that way, whatever the organism they are there, they will only grow in that conditions. The conditions were very, very selective. And if suppose similar kind of iron oxidizing bacterium is trying to grow, even if it is entering in the system, so like that we have kept the competition open. So same organism, the contaminant will also compete in the process. So in that way, whosoever will be fit to that conditions, so that organism will only grow. So in that way, we have kept non-sterile conditions, saving a huge cost of the process. Here you can see the ore samples which I have been for collecting. So this is a bauxite ore sample from the Radhanagiri, Kolhapur. See, now here the important point is we have talked about the bioremediation. Like, we have decided the problem of e-waste. Now the approach was bioremediation, biological entities we have to use. So, but where from we have to get the biological entities? Number one, as the people they says, the natural organisms which are there with the e-waste. But see, here the approach was totally different. Here we have the mechanism which we have chosen for bioremediation was the bioleaching. So for that we have to go for searching of the microorganisms. So this is a search for microorganism. So we have got this ore sample. So therefrom, and this is a Chitradurga, Karnataka pyrite sample. So these are the stones which contains microorganisms. So we have we have grown these microorganisms in our lab, and we have enriched, as I said, and these two enriched samples, like from the pyrite, from the bauxite, we have enriched separately, and then we have mixed together so as to get the mixed bacterial consortium. Fine. So this is what we have got, and we have kept the competition open, as I said earlier. This you can see the dark reddish color is nothing but it is indication of the good growth of the organism. So this is microscopic view. See, these are the circuit boards. E-waste. Here we have initially make it pulverized, and these are the results. Like, what are the metal content? As you know that metals, heavy metals, they are the more problematic agent. So we have analyzed it for the presence of heavy metals. So you can see that copper is there, zinc, lead, nickel, even some precious metals. Sometimes they are also present. So like that, the composition have been determined. Later on, we come to know that key, rather than using a pulverized form, non-pulverized form of the PCBs is more useful. And then we have stuck to the non-pulverized form of the PCBs. This is what we can say the the process. So this is a circuit board, large basis of circuit board. So the green color layer is there, it has been removed. Then it has been broken down into some small pieces, 4 into 2.5 centimeter. Then these pieces, see, this shining metal have been now removed. You can see only the notches, but not the metals. Is it? You can see that. So this metal removal has been done only because of the microorganisms. So it means that how bioremediation is going over here. So we are removing the metals from the e-waste, thereby. So why we should remove the metals from the e-waste? Thereby preventing the pollution. Pollution, which pollution? The groundwater pollution, air pollution, like that. So this is also called as a bioremediation approach. If you want to see the mechanism, see, as I said, this is an iron oxidizing bacteria. So iron is the energy source. So iron is giving electron. Once it gives the electron, Fe2 is converted into Fe3 plus. Okay, and now this Fe3 plus, see the bus lines. If you can see that, this shining, these are the bus lines. These are the bus lines. Bus lines, they are made up of copper. Okay, so let's see that this copper is in a Cu0 oxidation state. Okay. Now, whatever the Fe2 has been converted into Fe3, so this Fe3, it will oxidize the copper. It will oxidize the Cu0. And then, once it is oxidizing the Cu0, it is itself is converted into Fe2 plus. Whenever Fe3 has to oxidize the Cu0, it will convert it into Fe2 plus. It will itself get reduced, further be utilized for the growth of oxidants. This is Acidithiobacillus oxidans, the bacterium. What does it mean? It means that, see, organism is converting Fe2 to Fe3, and this Fe3, while oxidizing the metal, itself it gets reduced to Fe2. Fe2 is further available for the growth of. So, like as we have discussed in the last point, the consortium built up was different in generally how the consortium is built up. The typical way of consortium build up is taking separate to get a consortium. Okay, so that was a bottom up approach of consortium build up. Okay, now the top down approach, you should have a consortium from your natural habitat, and that consortium you're directly applying to the waste which you want to bioremediate. Okay, so this is the difference. How the consortium is built up. In first case, bottom up approach, wherein you are taking individual isolates, mixing it together, using it. In top down approach, you are using the natural consortium and allowing the consortium to get screened, to get select, depending on the reaction. Okay, this is a consortium built up, top down approach. So wherever you have to go for the bioremediation, I would suggest to go with the top down approach. Non-sterile conditions. Here in our case, we have used non-sterile conditions. The idea behind that is since it is a completely acidic medium, and the pH is around 1 to 2, so no organisms can contaminate that or that particular conditions. And the other thing is there is no any organic content in that media, so it is having only, we can say, the inorganic components, and so whatever organisms they are iron oxidizers, they will only survive in that particular condition. Let us say here also, if suppose from air any iron oxidizing bacteria is entering in the system, so it is welcome. There is an open competition. There is an open competition. So whatever organisms they are entering, we are allowing them to grow, even the contaminants. And during the process, whatever the organisms they are best fitted to the conditions, they will only be surviving. So this is how we did this bioremediation this thing. Here, as you can see that the organisms for bioremediation, we want to go for the bioremediation of the e-waste. So where from we should bring the organisms? So iron oxidizing bacterium, they are the native organisms, naturally occurring organisms at the ore samples. So these ore samples, we have brought one from the Radhanagari, Kolhapur, the bauxite ore sample, and then the second is from the Chitradurga, the pyrite ore sample. So what we did is we have uh, enriched. Basically, we have taken these ore samples, added in the media, whatever organisms they are there, we have allowed all of them to grow in a broth, and then we have mixed them together, the pyrite and the bauxite samples, we have mixed them together so that all organisms they were there, and this is nothing but called as mixed bacterial consortium. This is a growth of the organisms. The dark brown color, you can see more the dark brown color, more is the growth of iron oxidizing bacterium. This is a microscopic view. You can see that the cocci-like structures, and this is a jarosite. Jarosite is a precipitate. These are the circuit boards, the waste material which we have used. This is a powder of circuit board, the pulverized form. These are the compositions. You can see that the toxic metals, copper, zinc, lead, nickel, they are present. Even some precious metals, they are also present. Here in our process, we come to use than the pulver. This has a advantage. So this board, so exposing the copper, the bus lines, the lines which are there, the shining lines, these are the copper, and this is called as bus lines. You can see this is a small piece of the circuit board. You can see the shiny metal. This is the copper. Okay. So now, after leaching, after bio-leaching, all these shiny metals they have been removed from this part. You can see the notches, but not the metals. So this is the process we have. Then here comes, as I said, iron oxidizing bacterium, they are present over here. So iron oxidizing bacterium, they are oxidizing the iron. Fe2, they are oxidizing, they are taking electron from the ferrous, converting into the ferric. Okay, now it is a ferric which is reacting, which is oxidizing the copper, the bus line copper. Okay, so it is in zero oxidation state. So whenever the ferric is oxidizing the copper, ferric itself it gets reduced. Ferric gets reduced to ferrous. It means the copper which is in a zero oxidation state will be converted into Cu2 plus. Cu2 plus form, as we all know, it is a water soluble form. So Cu2 plus form is like copper is in a soluble form, and Fe3 is again converted into Fe2, the ferrous. So ferrous is again regenerated. Ferrous is again regenerated. So in that way, uh, we can say the food for the organism is regenerated. This is the beauty of the technology. This is the beauty of the technology. Um, so here you can see the growth of the organism on the plates. So here you can see that it is difficult to grow the iron oxidizing organisms on solid medium, uh, which we have got, and uh, uh, see dark brown color colonies. The dark brown color colonies you can see over here, these are nothing but the iron oxidizers. Zero oxidation state, right? Metallic copper ला आपण काय म्हणतो? We are calling it as a zero oxidation state. So ferric ion is oxidizing this copper. Thereby, the copper will be converted into Cu2 plus. Now, Cu2 plus, the form of copper in the copper sulfate even, it is Cu2 plus, right? So the Cu0 is converted into Cu2 plus. Fine. And now the ferric itself, it is converted into ferrous. Ferric is converted into ferrous. It means ferrous, which is a food for the or a bacterium, it is regenerated. It is regenerated. So this is the beauty of the technology. This is the beauty of the technology. The food for the organism is regenerated. Fine. So thereby reducing the cost of the process to a greater extent. These are the organisms, these are the colonies, as I said 2 to 3 times, though the slide was not visible to you people, that these are the colonies of the iron oxidizing bacterium. Okay. Um the reddish colored colonies you can see over here. As I said, it is very difficult to cultivate the iron oxidizing bacteria. These are not a traditional bacterium. It is very, very difficult to, first of all, to prepare a medium for the iron oxidizing bacterium. Number one. Number two, to get a colony of iron oxidizing bacteria. So these are, you can see the different types. These are the completely iron bacterium. These are the heterotrophic iron oxidizing bacterium. These are the acidophilic heterotrophs. See, these are the microorganisms which are playing an important role in the bioremediation process of this e-waste. To identify, we have gone with the DGT, density gradient electrophoresis, wherein we come to know that Acidithiobacillus is the dominating organism. We have also gone with the species-specific primers. And we come to know that there is a presence of Leptospirillum, the Acidithiobacillus ferroxidans, Sulfobacillus. These are some kind of publications on the process, as you can see over here. The bioremediation concept. Okay. But see, publication is not the end. Rather, before going to actually, there is a thought you might be aware of. Publish and perish, whether to go with publish and perish or whether to patent and prosper. This is a thought generally you might have encountered this thought many of the times. But what we did is with this bioremediation technology, the e-waste particularly, we have got the publications as well, but we have not restricted ourselves to the publications. We have thought to go with the technology commercialization. So success, we have got a success at laboratory scale. It was basically at TRL4, technology readiness level 4. So then we have thought to go with the scale-up studies. So scale-up studies were needed for the possible tech commercialization. So for that scale-up studies, we have approached the BIRAC, and so then BIRAC has supported the scale-up from from the flask level to the 10 liter as well as to the 100 liter scale. So whatever the bioremediation we have demonstrated at flask level, we should demonstrate at 10 liter as well as to 100 liter, so as to go to the more higher TRL. So we have thought to go with the TRL 7. So the problem, as I said, electronic waste, the hazardous metals in the electronic waste was the problem, and the solution, biological solution, microbial solution, we have given the remediation by using bacterium. And now this is a final technology. You can see the difference between the earlier picture and this picture. Here you can see this is a board. Okay, please see this keenly. This is a board, and it has this shining silvery metals. Okay, so this is nothing but a solder. So for the first step, we have selectively removed this solder. You can see the board without solder. Okay, so this is pre-leaching, removal of only lead and tin. After that, we have removed the green layer. That is after pre-treatment, we have got the board like this. So all the shiny metals, they have now exposed. Why to remove this green layer? Because with the green layer, bacteria cannot access the metals. Okay, so we have selectively removed the green layer. This is after pre-treatment. And now we have, we have dipped this in a biological container for the bioleaching. And after bioleaching, see all these shiny metals. The notches are there, but the metals have been clearly removed. So this is a board we have obtained. And see, whatever the metals they are there in the liquid form, they can be recovered by electrodeposition. And now the remaining liquid, it can be regenerated to get the leachant. Okay, as I said, the beauty of the technology is regeneration. So it can be regenerated for the next step. Okay, So this is what we can say the technology we have developed. Finally, this technology we have developed, and the novelty, as you can see that metal removal from the waste printed circuit board. This is a novel method. Non-obviousness, even though it is a microbiological process, but since the organisms they have been used, they are not the typical one. That's why it is not obvious for any microbiologist to do this particular step. Then inventive step, structural form of the e-waste, type of biological and non-biological solutions, sequence of their application. This is nothing but the inventive step. Utility is any eco-friendly metal removal. Basically, it is if any company is following the metal removal from the e-waste, so they should adapt this technology. So these are the efficiencies obtained. Around 99.99% of the copper and 93% of zinc has been leached out from the BCBs. See the bioremediation efficiency. Copper recovery after electroporation it was 92%. As I said, this one. The optimization. See, these are the 10-liter scale studies. Um, all these flasks, these are the water jacketed reactors, and when the temperature it can be automatically adjusted. There is a robotic motor as well here, and here you can these are the optimized parameters for the process. So you can see over here, and on the basis of these optimization studies, the 100-liter, these are the 100-liter scale demonstrations. So we have in our campus, in our department, we have these 100-liter scale reactors ready for this complete technology demonstration at 100-liter scale. These are some economics, every step, what the cost is involved. So accordingly, it has been calculated, and the process cost was estimated around 894. But if you are using a solar distilled water, it will be minimized to 586 rupees. This is a value, total value of metals. See, once we are extracting the metals, it has some value. So that value which has been calculated, it was around say 914 something. So you can see that these are the statistics, or so these are the final economics, conclusive economics. And for each cycle, around 2.5 kg of PCBs can be processed. As you know, there is a huge market. See, e-waste is continuously increasing day by day with the increase in technology development. So the market for e-waste tech management is huge. So TRL, this TRL is right now at 7, TRL 7, late stage validation and pilot scale demonstration of technology at 100-liter scale. So now this technology is ready for TRL 8. Yes, so BIRAC path patenting has been facilitated. Actually, yesterday was 11. Yeah, day before yesterday, the patent for this, the complete specification have been filed by the BIRAC for us. So it was an 11th. So this you can see that the date is 12th. It was a priority application, and that 11th is a date for the complete specification, exactly before completion of the 1 year. Fine. And the technology transfer is also being facilitated by the BIRAC, Government of India, under the BIRAC scheme. So very soon we will be getting the technology transferred. These are the cognizances by the media for this bioremediation technology. See, all of this, why we are discussing today, these are the actual practical demonstrations for bioremediation, which we did here. That's why your teacher has thought key for the PhD students, it would be very, very useful to have some case studies done around. Yeah, apart from that, this is just for giving an idea. We are discussing bioremediation. So, whatever in our college campus, whatever the institutions you are in, so there is leaf litters. Pala pachwa jala म्हणतो आपण. So the all leaf litters they are there. What we are doing with that? Any bioremediation option we are looking for. So simple. These are the drums. We just have to put different holes and make different holes. This drums, we just have to put the plantlets here. Okay, we just have to put the plantlets here. No soil at all. No soil at all. Okay, simple plantlets. We have to put it through these holes, and then we have to simply add the leaf litters in these drums. We have to add the leaf litters. पालापाचोळा जो काही आहे कॅम्पस मध्ये कॉलेजच्या तो टाकून दिला आपण ह्या ड्रम मध्ये. Okay, so and we just have to sprinkle the water. So every day we have to do all these things. So what will happen, all these leaf litters, it will slowly it will be decomposed naturally, and that decomposed material will be a good nutrient for the plant growth, will be a good nutrient for the plant growth. You can see this picture and now this picture. So it has flourished in a very good way, so that even you can see, you can't see the drum behind, you can't see the drum behind. Okay, it has flourished in that way. So basically it is all because of the leaf litters in our college campus. So this is nothing but also the remediation. This is a spirulina. Actually, I should not discuss. I don't know, but yes, we can use the spirulina, or we can say any microalgae. This is basically microalgae cultivation. Microalgae you can also use for bioremediation options. So here in our college campus, we have a corridor and there is a huge space. We have a huge gallery. So there we have placed these pipes. Okay, and these pipes, they are having the running this thing, what we can say, microalgae. So they're utilizing the free sunlight as well as free CO2, and the growth, microalgal growth is there. So though it is not that much related to your today's topic, but as a part of discussion for the cultivation of microalgae, I have shown this. So this can be there. Yes, and now we are ending our lecture with this statement. Anyone can answer this? Okay, what exactly I mean here? Yes, anyone can start. Anyone can try to tell this the meaning. Okay, thank God. So yes, who can tell me the meaning of this statement?
You can unmute the mic. Sumane madam.
Sir, I think it means microbes can be everywhere, but we can't be everywhere. Like, even we can predict that
That's
they could
true.
be there in the space, but
Okay,
we
okay.
might not be able to reach planets where they might be there.
Okay. Anyone else? Actually, it's true. The meaning is like that. But by sentence, I just want to read again. So there are almost no places humans can go, but microbes can't. So there are no such places where humans have the ability to go, and microbes, they don't have ability to go. Okay, there are almost no places like that, but there are many places where microbes have ability to go, to wander over there, and but humans, they don't even think about going over there. Okay, it means the bottom line of this statement is microbes have, microbes have a huge potential and a enormous ability to, there is a huge physiological diversity as well, so that the humans should utilize the potential of microorganisms. you know uh, it is our limitation, it is human limitation, that we can't recognize the potential, the role of microorganism at such places, and we fail to apply in that particular sense. Taking an example, like, see, iron oxidizing bacterium, where they live in mining environment. So if we have a problem of e-waste, so then we just have to think, key iron oxidizing bacteria from the mining environment, we can utilize that organism for the benefit of the human being, thereby removing the metals from the e-waste. So how will come to know that this can be applied to that? So we will come to know once we try to study that. Once we are knowing them, so then obviously we can utilize, we can exploit them for the application. Okay, so for that, we should explore the microbial diversity as much as possible. So till date, as I know, that very minute fraction of organisms, microorganisms, they are being known and they can be cultivated, but there is a huge fraction of the organisms which are still uncultivable, and people, they don't know even about that. So if we try to explore the diversity, microbial diversity, definitely, there would not be any problem on Earth which is remain unsolved, because it is the ability of the microorganisms. And the statement, never underestimate the power of microorganism, this is a truth statement. This is the original statement. Later on, the statement has been utilized in a one movie for another some instance, but the basic statement given by the Louis Pasteur, it is never underestimate the power of microorganism. Okay, this is actually the statement is. And definitely we will get a remedy for each of such kind of problem with the microorganisms. Fine. Microorganism. Okay, this is actually the statement is. And definitely we will get a remedy for each of such kind of problem with the microorganisms. Fine. So with this, I think I should stop here. And we are now actually we are keeping the forum open for the discussion. Definitely you raise your questions, you raise your queries, so that I will surely try to answer that. Fine, you can. Yes, any queries, any questions?
Sir, I had a question.
Yes.
Sir, so we use these microorganisms to remove metals from
Hmm.
e-waste and they
Yeah.
grow.
Hmm.
So what to do with those microorganisms after they grow?
Mm, yes,
Like
they are
how,
continuous. Huh.
like they have increased in their culture, they are growing in number and biomass. So
Okay.
what do you do with them later on?
Basically, if they are more and more in numbers, so they are more beneficial to the next round of leaching. You're getting my point? Next
Yeah.
round
So
of leaching means what? Continuously, see, the e-waste is in a huge amount. So
yeah,
we continuously need the huge amount of microorganisms. So if they are continuously growing in our defined culture media, if they are continuously growing and they are in a more number, so more iron they will oxidize.
treatment is possible. But then, sir, you will have to maintain this for a very long period of time, continuously.
Yes.
Those
So
cells.
continuously, see, whenever we are talking about the commercial scale plant, so every day
Hmm.
you are requiring this, no?
Hmm. Hmm. Hmm.
We are thinking from that commercial point of view. Every day, obviously, you have to require. Rather, if you are not working your, if your plant is not working for a few days, it would be more difficult to maintain them, because they are very, where they are very tricky. It is very tricky to grow them and to maintain them.
Okay. Okay.
So it would be always beneficial to have the organisms in a greater number, and to get it continuously grown.
Okay. But then, sir, culture will be reaching at some point to saturation of the growth where
Uh,
the number is too much or too high. What do you do in such situation then?
uh, no, actually, that's what I'm saying. Whenever we are talking about, see, even at 100-liter scale, so there is no
Hmm.
any point we have obtained where we are having a problem of the growth itself. Rather,
Okay.
actually, it is having a very low generation time, and we have to wait for months to. I'm
Oh.
talking about enrichment. I'm talking about enrichment. Huh?
Hmm.
If
Hmm.
suppose,
Hmm.
say for example, if my plant is not working for a few days, okay, and say 1 or 2 months, say for example, and then I need a culture. So to get a active culture, I have to wait for 1 or 2 months even to get
Oh.
active culture, to get, because its
Okay.
generation time is more.
Hmm. Hmm. Hmm.
Yeah, but once it is in an active phase, once we are utilizing it every day, so then
Hmm.
even though the organism's growth is faster, even the organism's number is more, it is not
Hmm.
creating any problem. It rather
Okay.
it's
Okay.
beneficial, because more the number of organisms that are there, more fastly the ferrous will be oxidized. More fastly the
Okay.
ferrous will be oxidized, more quickly you will get the ferric leachant. More quickly you will getting the ferric, so the process time it will be minimized, if suppose in that way.
Yeah. So getting rid of microorganism is not a problem
It's
because
not a
it
problem as
never
such.
happens.
Yeah, at
Hmm.
least we have not encountered with that.
Okay. Sir, so far in our labs, we have only seen E. coli growing.
Yeah, that's what. So
It grows so fast. So I
Yes,
had this
that's what
doubt
I'm
in
saying.
my
It
mind.
is very easy to grow the chemoheterotrophic. E. coli is a chemoheterotrophic organism.
Yes, yes.
It is very easy. It is very typical, and it growing this bacterium is very very tricky, and it is very we really need a huge patience for to get
Hmm.
out of it, even in the broth or in even in the solid form.
Thank you, sir.
Yeah. Okay.